Good afternoon, my name is Gabriela Zamora and I'm going to be an upcoming senior at Lane Tech. This year I worked in the ecology lab with Paul and Dan. The title of my project is The Relationships Between Nutrients and Biomass Production in Wetland Mesocosms. Some background research. Wetlands are a vital part of our Earth's ecosystem. They serve as a source to stabilize water supplies, clean polluted water, protect shorelands against flood that goes into the land. Because wetlands sustain such a rich biodiversity and provide a great amount of habitat, scientists are really interested in studying and preserving our wetlands and our environment. For the past 15 years, humans have doubled the amount of nitrogen that passes through the nitrogen cycle through fossil fuels, crops, and other purposes. So I was interested in looking at the relationships between nutrients and biomass production by a native wetland plant, Mesocosms, to see if nutrient availability influences the biomass production. So Mesocosms are controlled mini wetlands at the University of Wisconsin, and those are the ones we used in this experiment to investigate this relationship. I had two questions. The first question was, does below ground root biomass correlate to above ground biomass measures? And the second question was, does biomass correlate with nutrient availability, nitrogen, ammonium, and phosphorus? So my first hypothesis was that Mesocosms that have a higher above ground biomass production will have a lower below ground biomass production. And the second hypothesis was Mesocosms that have a higher background nutrient concentration in the soil will have a higher overall biomass production. The major materials were obviously the Mesocosm, the soil, the biomass, and the root and the shoot biomass. The rest of the materials will be mentioned in the experimental procedure. So we had to do KCl extractions in the soil nutrients. The first step was to make the 2 molar KCl solution. Then we had to homogenize the collected soil from the Mesocosms by smushing it around. And we had to weigh out the soil samples and put it in the drying oven so we can get the soil moisture out of the soil samples for the Mesocosms. Then we had to weigh out an additional soil samples and put them in the test tubes so we can get the KCl extractions from them so we can get the solution for the nutrient analysis. And once we had the KCl extractions, we put them in that awesome machine down there, which is the CLAQ2+, and it runs the nutrient concentrations, and we ran a standard procedure, and it produces a color to calculate the concentrations of nitrate, ammonium, and phosphorus on the soil. So these are the results. This is what we looked at the relationships. The first figure shows the shoot biomass and the stem height, and there's no correlation. The second figure shows no correlation between the shoot biomass and the root biomass. Figure 3 shows a positive correlation, but it is not significant between the shoot biomass and the ammonium concentration. Figure 4 shows no correlation between the shoot biomass and the nitrate concentration. Figure 5 shows no correlation between shoot biomass and the phosphorus concentration. Figure 6 shows no correlation between the root biomass and the ammonium concentration. Figure 7 shows no correlation between the root biomass and the nitrate concentration. And the final figure, figure 8, shows no correlation between root biomass and phosphorus concentration. So in conclusion, both of my hypotheses were not supported. The data showed that only the shoot biomass and the ammonium concentration had a positive correlation, but it was not significant. And the rest of the data showed no correlations, therefore none of the nutrients seemed to relate to the biomass production in the Mesocosms. This could have been due to the small sample size, and also the treatment that the Mesocosms received had been the same for the past 5 years. So some of the ways to improve this project, obviously, is to increase the sample size and to test biomass production under different nutrient treatments. So the big picture. Wetlands are extremely beneficial to humans. They filter harmful substances, protect against floods, protect against erosion, and provide food and habitat. So due to their importance, it is really critical that scientists and all of us work to understand how to preserve wetlands. That's it. I would like to thank everyone who helped me with this project, especially Laura. She was an awesome RU intern, and we try not to blow up the lab. And Paul, he was awesome too. And thank you, Ben and Murphy and Shelly and all of you guys in College First. questions? Yes.
What did you mean when you said they had been receiving the same treatment for the last 5 years?
So pretty much the Mesocosms are like little tiny plants. It wasn't actually an actual wetland where we just went out and got soil samples. So they've been sitting there for 5 years with the same type of weather and no like treatment to it. So that's what I meant by that.
I'm sorry, I didn't get the treatment. What were you adding? Fertilizer or what else?
Well, I actually didn't do the field work, but there are like mini little plants with soil in them, and basically they're just out there in the field and they have I don't know if they have any fertilizer or anything, but they all have been sitting there without being touched for 5 years. Yeah. Okay, thanks. Oh.
Do you think that if you tried this experiment in different ecosystems, like if you tried it in a prairie, if you tried it in woodlands, do you think you'd find similar results or different results?
Yes, because varying on the environment, maybe one wetland has more like living organisms in there that would affect the productivity of like the above ground and the below ground, which it would vary between like a place where it's really isolated and there's really no like high biomass. Yeah.
Do you think if you did this again in like a native wetland habitat that wasn't a controlled like group of plants, that you would get the same results, or do you think your hypothesis would be supported?
I think my hypotheses would be supported, but I'm not sure because if we tested on just one wetland that was native, yeah, then we would have like maybe like a correlation in between. But since wetlands vary so much from like each other, even if they were native, depending on where they are, so it's kind of hard. That's why we're studying still wetlands.